So hi everyone. Thank you so much for having me. I'm really looking forward to hearing the other talks as well by the other INS practitioners. As Callum said, my name's Emily Iles, and I've been working for Tweed Forum since 2015. And mostly I manage the Tweed Invasives project. So in this webinar, I'm going to be talking about how we've been tackling Himalayan balsam by using classical biological control. I will do my best to field any questions at the end, but please be aware that I'm not a plant or pathogen scientist. So if there are any really technical questions, then just email me and I'll get one of the scientists from CABI to field that one. And I'm trying to figure out how to... There we go. Technical issues. So I just want to give you a quick bit of background about the Tweed Invasives project as a whole. So it began in 2002 after a strong public demand for some action to be done, as much of the catchment was dominated by the INS that are on the left. So giant hogweed, Japanese knotweed, and we have some instances of American skunk cabbage, but it's not too bad. So we're now at year 19. So giant hogweed and Japanese knotweed have been systematically controlled each year by spot spraying using a licensed herbicide. So we have trialled other methods in recognition that herbicide is not always the best method. We have found that it is probably the most effective for these species alone. We are at a really exciting point in the project whereby there's been huge dramatic changes in the catchment and the abundance of these INS. So we're now at a stage where we're not controlling large swathes of these INS, we're more focusing on biosecurity and keeping these species out, and also horizon scanning and thinking out ways of, including more volunteer work and that sort of thing. So we will probably continue to use herbicide for those particular species because our usage has dramatically decreased. So our management goal on those species is eradication, if possible. And again, this requires us to systematically target every single individual plant. And this is no mean feat, as our catchment's 5,000 square kilometres. So it's a bit like having a huge unruly garden. You don't really know where everything is. But we've also come to realise that Himalayan balsam requires a completely different management approach. And hence I'm going to speak about this. So we've all seen it when out walking. It has this beautiful pink flower in the summer months, and it has these charismatic popping seed heads in the autumn. It is a plant native to the foothills of the Himalayas in Pakistan and India, and it grows there in mixed populations with other native plants there. And there it doesn't dominate its habitat. So it was introduced into the UK as a garden ornamental in sort of the mid-1830s. And because it has no natural enemies in the UK, it's become very extensively widespread, as we all know, in the UK and in Europe, and it's causing problems. So some of these problems include its ability to outcompete native plants for space and light and resources. We've also read recent studies that have shown that where there are dense populations of Himalayan balsam, the local invertebrate population and biodiversity is much, much lower than on a site containing mixed native species. So this can lead to fragmented, destabilized ecosystems. And this has serious consequences on processes and normal functioning of ecosystems. So the flowers of the plant have the highest sugar nectar production per flower of any native European plant species. So this enables the plant to attract numerous pollinating insects away from them, which also prevents native flowers from becoming pollinated. And each plant is very successful at reproduction, so they can produce numerous seeds which are flung via the seed pods about 7 meters away from the parent plant. Research also points to Himalayan balsam causing flooding and soil erosion when plant material dies back in the winter. However, I guess it's not all doom and gloom. So we're lucky, the plants, unlike giant hogweed, it doesn't harm humans or animals. And at least it does provide nectar for bees, which we know are in decline. So essentially the problem is that this species is a problem at high abundance. So we need to figure out a management technique that can reduce its abundance. So we can tolerate it at a small level, but we need to figure out how to stop it from becoming a monoculture. So we've tried lots of different approaches, which I'm sure you'll have seen before. The method we've tried in the past to reduce abundance in dense areas, mechanical methods, so strimming, cutting, hand pulling. Some of these methods are beneficial. You can get volunteers involved. It's a good education tool. It's quite a fun day out. However, it's not a long-term management measure, and it can only be done on a small scale, which means there's the likelihood of recolonization. So if you want to do that year on year on year for the next 25 years, then that's going to be the case, unfortunately. We've also tried chemical application, and we've also figured out that you can lose a lot less concentration of the chemical. The plant really doesn't like it. However, again, we're using a broad spectrum herbicide, and much of the Tweed catchment is SSSI and SAC, and we just really don't want to do that. Again, if you're not going to eradicate the species by using this method, then you're basically committing yourself to year-on-year herbicide application. So we're trying to come up with a longer-term solution that's much more sustainable and doesn't use as much herbicide application. So in an effort to do this, we have come up with trialing the classical biological control. So alongside CABI, so the Centre of Agriculture and Biosciences International, the ultimate aim of this project is to introduce a co-evolved natural enemy that exclusively attacks Himalayan balsam whilst leaving indigenous species intact so that the ecosystem can be restored. So the rust pathogen, with a very complicated scientific name, was selected by CABI as the best candidate for classical biological control. And it was brought over to the UK for testing in 2006. So there are many concerns about biological control. Cane toad springs to mind. However, there have been lots of advances in scientific approaches and biological control. And rust funguses have a really good history of use in biological control because they're very, very host specific. They've co-evolved with their host species for thousands of years, and so they are considered relatively safe or very safe. So the species, once it was in the UK, it followed extensive testing, and it was approved for release by DEFRA in 2014. So we got involved in 2015, and the initial releases involved us planting out an infected individual plant or a couple of individual plants in an area of healthy Himalayan balsam plants. The rust fungus itself, which was selected, it has a very complicated life cycle, and it involves 5 different spore stages. It completes every single one of those spore stages on the host plant itself. And if anybody is particularly interested in the life cycle of the rust fungus, I've included a link to a video in the back of the presentation. It's from the CABI website. but I don't have time to go through it all now. So essentially, once the rust takes hold on the plant, it's got multiple attack mechanisms. So the first spore development is these tiny little orangey yellow spores that you can see, and these start on the seedling stem, and it basically makes the stem warp and bend, preventing growth. So plants end up being much more stunted than healthy plants. The next stage is the spore infection on the leaves of the adult plants, which appear as these sort of brown, dusty looking spots, or brown sort of powdery spots. If the infection is really high on the leaf itself, it can cover up to 70% of the underside of the leaf, which basically dramatically reduces the photosynthetic potential of the plant. Plants that are infected by the rust also have reduced seed production rate compared to healthy plants. And any damage done in the various life cycle of the plant means that the plant is much more susceptible to insect damage and other pathogens as well. So it becomes less healthy as well. And in order for the rust to be able to do this, essentially in the wild and become successful, we need to build up a really abundant population of the rust for it to reach a sufficient enough level for it to spread to other healthy plants in the environment. And this has essentially been our goal once the rust has been released. So we get a lot of interest in the project, and a lot of people are really want it. So we've got some landowners who are very interested in the project, and they say, can we go and release it straight away? It's not as easy as that. I'll talk about the different stages in our approach and the practicalities of hosting a site, etc. So prior to releasing it, there's a lot of hoops to jump through before having a rust site. And most of the activities on this page we do the year before. So initially the first stage, which is fairly obvious, is to gain landowner consent, which is sometimes easier said than done. And also government consent from the relative agency. The permissions and the licensing is different between England and Scotland as well. So tricky for us because we're cross-catchment. The rust itself, we also need to select the right rust. So it's a requirement for us to test which biotype of the plant is present at the site, because there's different types. So this is done by collecting seed samples the year before, and we send them to Cabby, and they do some genetic testing to determine which type of rust is going to be compatible and which one they'll send out to us. So in the picture, that's the little vial of the rust spores, which gets sent to us in a big package. So something that we have realised as well is that site selection is really, really crucial, and it's something we've been working on and developing and sort of adapting as the project's gone on. So since the first releases in 2015, and Cabby have been working really hard to optimise the release technique, and we've changed it every year as the project's gone on. And the type of environmental conditions have been studied, and basically the conclusions that have been drawn from that about optimum conditions required on the particular site in order to establish this really good population of rust, which I spoke about. So that we need the site to be not liable to being submerged by water for extended periods of time, which is easier said than done, as most of the Himalayan balsam is prolific in the riparian zone, which is liable to flooding. So it has been quite difficult to find a site that isn't. It must have like a dense population, which isn't a problem because we've got lots of it. And the site must be quite open, so it can't be in woodland or overly shaded. So, which is also quite tricky because a lot of the plants grow in sort of riparian woodland or sort of willow thickets and that type of thing. So site selection is quite tricky, that I found anyway. So once all of that is established, once you've got the site and all the permissions are in place, and we know that the site's going to be good for hosting the site, etc. Cabby send out a big box with all the equipment that we need for the releases during the summer, and a programme, sort of a timetable of when we need to conduct all the activities, which is very handy. So firstly, within my site that I've selected, I mark out a site with canes or rope or something like that, tape, that's quite visible for me, but not overly visible, because I have had a site that's attracted unwanted attention from somebody, from youth perhaps on a footpath, so I don't make it too visible for passers-by. So the spores from the label vial, which were in the picture before, these have to be kept in the freezer. Once I take them to site, they're opened and we put them in a bottle of water, spray bottle of water containing a surfactant, and this is enough to do about between 5 and 6 meters squared area. They are sprayed onto the undersides of the leaves because this is where the spores infect the plant, and the green box there highlights sort of the programme of monitoring. So the first inoculation occurs, and then about 5 weeks after that, we'll go back and do the monitoring. So in order to monitor the infection, the monitoring, again, the methodology has slightly changed over the years, but now what we do is take photos of lots of individual leaves, and we can supply them to Cabby either by an app or by just a Dropbox, that type of thing. And after that, they're sort of analysed by an image processing tool, and basically it will analyse the level of infection at the site, and also we have to analyse how far it's spread, if it's spread. And then the following year, we will do spring surveys on the seedlings to see if it has overwintered and infected the seedlings the year after. So the sites that we've done this year, I won't know how affected they've been until I've done the spring survey. So it's waiting time. I also put a data logger at each site, and this gathers environmental data. So temperature, humidity, that type of thing. So our results. So in the early trials, I think I mentioned earlier that initially we were given a handful of infected plants, which we had to plant out and keep alive. Not like me to try and keep a Himalayan balsam plant alive, but we were desperate to, in a really hot summer. We found it really difficult for these plants to spread their infection to other plants, I think probably because there wasn't a high enough inoculation rate in the population. So that methodology got changed, and laterally Cabby managed to get permission so that the inoculum via the spray bottle could be used, and we could spray out a large quantity of the rust onto our 5 by 5 meter plot, and we could therefore allowed us to get the better coverage, and so more plants could be infected. So in sort of between 2017 and 2019, our Himalayan balsam sites were successfully infected with the rust in the summer. But the following year, when we came to do the seedling monitoring, we were struggling to find any seedling infection. And I think this was probably due to us not realising how important it was for the environmental conditions to be just so. So I think some of the seedlings had possibly got damaged during water being there in the winter time. So we adjusted that. In 2020, we managed to get funding for 3 sites in Scotland and 4 in England. it's the first time that any rust had been applied on sites in Scotland. All of the sites were successfully infected. And then at 2 sites, we conducted our seedling surveys this year, and we found the yellow spores in the picture to the right, which was really exciting. So we know that the rust can survive in Scotland, in the Tweed catchment. So that was our main sort of first hurdle, because we weren't sure whether the rust could survive the winter and go on to infect seedlings. So we know that is the case now. And also at two of the sites, the spores had progressed onto the adult stage, and we saw infected adult leaves, and it also had transferred onto other adult plants. So we're getting really good results of it, not only surviving the winter, but going on to the next stage and then spreading to other plants within quite a small area. However, I know that Cabby have had really good results down in Cornwall, where the rust has been spreading up to like 100 meters or so. So that is really exciting. So it has the potential to do that. So this year I've inoculated 4 sites on the English side of the Tweed catchment, on some of the tributaries on the River Tail. So the results are still being analysed, because I only submitted them sort of October time. In the summer, they had successfully infected Himalayan balsam plants, and we saw a spread of about 8 meters, which is really, really promising. And they were infecting other healthy plants. And the rust infection appeared to increase over the summer. So not only the plants that I first inoculated, the first monitoring showed like a decent infection. The second monitoring, it was really evident. So that's great. So our next steps, we are really encouraged by the results. It has taken a long time and a lot of effort, and probably quite a lot of money as well. But I think we are going to continue with it. We're definitely going to continue with it, because it seems to be our the only thing really that we're enthused by. We've still got to do our other assessments on our sites this year, which will take place in spring 2022, and we're going to be looking for new sites next year for the following year, so it'll be 2023. So I've put together a list of useful information for everybody. So I know that this slide is going to be available. Please have a look at the Cabby website. As I said earlier, there's a video about the life cycle of the plant. They also have a dedicated Himalayan Balsam project page, which has a collection of papers and articles, which could be really useful. There's also a link on other species of plants which are currently under biological control. And there's also an insect which is being used to attack Japanese knotweed, which is really interesting. There's also information about our project as well, over the history of the project as well. So thank you very much for listening, and I'm happy to take any questions. I will now stop sharing my screen.
Okay, thanks, Emily.
No worries.
Well done, pretty much to time as well. Fantastic. We've got a few questions coming through. We'll try and sort of put them through. I think some people I think we're all hunting for a silver bullet here, but there's 2 or 3 questions, I think, particularly about the environmental conditions. I think you mentioned that you're recording. Yeah. So I'll just run through those in the order pretty much that they've appeared, and see how we get on. First one's from Andrew Skinner, is about the inundation and the avoidance of flooding sites. And the question is that inundation avoidance during the establishment phase, or can we actually, once you've got the thing infected and established, can it work in these sort of locations, or is it a real showstopper wherever it's there?
Yeah, so as far as I'm aware, I'm pretty sure that it is just in the establishment phase. So as I said, we're looking to establish a really good population of rust. And at the minute, if we can't do that because it's being inundated by water, and it's preventing that the seedlings from being really, really infected, then we can't do that. So I think once we've got a subpopulation that's spreading and that's really, really infected, I think once it spreads to other areas that might get inundated, it shouldn't be a problem. But I think the CABI team are definitely doing a lot of research on that.
Yeah, okay, thank you. I'll come back to Andrew's second questions, because inevitably there's always a question about the money, isn't there? The next one's from Emma, is about, is there a hypothesis as to why the woodland or shaded areas don't show such good spread? And you mentioned that the tactics are to use open sites with little or no shade.
There is a paper on it. Yes, I think it's mostly due to humidity levels. But yeah, that's definitely a question for CABI, that one. I'm pretty sure if you go to the link, there's a list of scientific papers, and they have disclosed the environmental conditions. Yeah,
okay. We did have a question about the cost per 5 by 5 meters for the inoculum, and I think we've got a colleague, Jamie, I think from CABI, who's sort of sharing a little bit of information there about the cost of £8,000 per site, 3 inoculations per year. So hopefully that gives an indication, if nothing else. I don't know if Jamie wants to put anything else into that, other than, I guess, that in terms of what the cost would be for volunteer work, it's clearly higher than that, but maybe once it is established, I think the hope would be that it continues to run without cost, I guess.
Yeah, essentially that's what we're hoping. The initial project site is expensive, so for us it was circa £7,000 a site. But obviously what we're hoping is for the rust to establish, and then it will be self-sustaining.
Yeah.
So again, we are willing to take that initial cost hit if it means that we're coming up with a solution that is sustainable, and also potentially cheaper than throwing chemical at it or getting in help with hand pulling, etc., which we know doesn't work. So
yeah, and then we've got the fatalistic Mark Cubitt, which is almost the abandoned hope, all ye who enter here, I think. It's for areas that don't meet site selection criteria. Emily, is there any hope for the future?
Yeah, so I think, again, we're still in the trial phase. It is still a biological controlled trial. We are still trying to establish a really good population in an area that meets all these criteria. But I think once we have established a really good population and it starts to spread, I don't think the site characteristics, the ideal sort of site characteristics, are going to be so much of a clincher. But as I said, there is a lot of information on the CABI website, and I know they've done extensive trials on environmental characteristics. So I think go and have a read of that, and it should hopefully answer any questions.
Yeah, okay. I know also we've had a little bit of problem with some people getting into the presentation earlier. I think that's reflected in the question from Stuart Pritchard about does the rust eradicate balsam. Just to confirm to Stuart and others that were unable to get in, the whole session has been recorded this afternoon, so you'll be able to view the whole presentations on the website once we tidy things up and put it up. I think the question is that the answer to Stuart's question is that at the minute, we're still, Emily and colleagues in Tweed are working through sort of trial procedures to identify best approaches and methodologies to get the rust established in their correct places. So at the minute, it doesn't feel like an eradication option right now. It's still a sort of a refinement of process, Emily. That'd be reasonable.
Yeah, and also earlier on in the talk, I mentioned that Himalayan balsam, it doesn't cause harm to humans. It does provide some late flowering nectar to pollinating insects. and it's only problematic when it's in a monoculture. So we can actually tolerate it at low levels. So we're not looking to eradicate it. We're just looking to prevent it becoming a monoculture. So this technique won't eradicate it. It will just prevent it from being able to outcompete native plants in a vast area.
Yeah, okay. We've got quite a few questions coming in. I'm conscious we've got another 2 presentations. Going to try and deal with a couple more, and then maybe we can invite Emily to look at the rest on the chat box and maybe respond to those while the next presentation's on. There's a question about, you mentioned the rust fungus being non-native to the UK, and I picked it up as well, but also that they're very host-specific, Emily. So are you anticipating that the rust is going to have any negative effects on the environment or other plants, or is that sort of risk pretty low down the pile, given these are, you know, host-specific rusts? Yeah.
So when the rust was brought in transferred over to the Cabby Labs in 2006, it went under a very, very rigorous testing program, and they included over 70 plants that were very closely related to it. And the rust does not want to live on any other plant other than Himalayan balsam. So it can't survive on other plants. And they went through years of testing and followed a very, very strict protocol. So I'm really confident that it is very host specific and it won't damage any other plants. But yeah. Yeah.
Okay, and then there's a couple of questions. One from Nicola about how far the rust is able to spread, and I think from that it's variable. You know, you're still measuring the rate of spread at sites, and Cabby are doing that in England as well. And then one from Harry about can the spores be collected and stored to reapply? And I think the hope is, from my understanding, is that you establish the spores within the population of balsam, and therefore they maintain themselves within that population, and you don't need to, you know, collect and then reissue. They're sort of established in the balsam plants themselves.
Yeah, so collecting the spores would be quite a time-consuming process because they're very, very tiny. And that's essentially what the guys do in the lab, is they brush off the individual spores from the plant and they post this up a vial which is this big, and it's taken them ages. But the spores themselves, like other species of plant and pathogen, they're windborne. So that's going to be how the spores will spread from plant to plant, is via the wind. Okay,
thanks so much. A couple of sort of observations really coming in. I think I'll probably try and round these up. And I think that, you know, you mentioned about all the other tactics that people use for balsam, whether it's glyphosate dosage or hand pooling, and, you know, the limited impact of those on sort of catchment scales. For me, this looks like as an observation at this point that we're really trying to identify other options for control, and the rust fungus may well be something that can be applied to within catchments or maybe in specific locations, but it gives us an option for control which maybe can be nested alongside these other things and makes the opportunity for containment or potentially eradication potentially reachable, where you have a portfolio of methods that are all in play and not absolutely relying on one or t'other to get there. Anyway, do post further questions on there about this one, and Emily will come back to those. But I'm going to say, Emily, thanks so much for that. Obviously, we were not surprised about the interest in that presentation, and we're really pleased that you were able to come on and join us. But if we can invite you to mute and hide, if you like to do that, I'm going to introduce our next speaker. Our next speaker is Charles Hughes from the Canal Rivers Trust, and he's running a project Precept, which has got eradication in it, which is quite a bold statement for a project objective. It's a new project running 2021 to 2025, but I spotted Charles's posts on Twitter and thought there was some really quirky stuff going on here with big machines and also some weevils. So poor Charles got a sort of cold call from me to see if we could actually persuade him to give us a little bit of a feel for what the Canal and Rivers Trust are doing, and we're particularly interested in mechanical control of floating Pennywort, and also he's going to talk to us about biocontrol using weevils to control the floating water fern. So Charles, the floor is yours. Hopefully your slides will appear as if by magic any second. And I think my waffling on is just busking you some time for that to happen.
Great, thanks very much, Callum.
Just before you start, I sort of mentioned it in the passing. Apologies to those that were locked out or were unable to gain access initially. We're pleased to see that the head count is now bounced around north of 60. So thanks very much everyone for your patience and tolerance in getting into the thing. And just to reconfirm, all the presentations will be recorded and will be available on the website, and we'll make sure that you receive a link or are able to find those after we go. So apologies for any hassle caused by the sort of locked bolts to the meeting, but we're glad that you've been able to get in now. Without further ado, Charles, the floor is yours. I shall shut up.
Thanks, Callum. Can I just confirm that the presentation is visible to your?
Yes, it is.
Great, that's perfect. Yeah, thanks very much, Callum. And yeah, fascinating first talk to follow on from. Really interesting to see what other organisations are doing. But from my perspective, I'm Charles Hughes. I'm sort of the invasive species project manager at the Canal Rivers Trust, but I'm also one of the environmental scientists working on sort of contaminated land and water quality issues as well. So a few different aspects of environmental field that I try to get involved with. And today, really, I'm just going to talk about a project, as Callum said, that's begun this year in partnership with Severn Trent Water. And today it covers a number of different aspects, the project, but today we're going to focus on the aquatic side and look at some of the treatment methodologies that we've been undertaking, very much on the practical side of trialing new techniques and also trying to improve existing ones as well. So thank you for joining. It's just a bit of background, really. The Canal Rivers Trust, we're responsible for around 2,000 miles of waterways across England and Wales. I predominantly work within the West Midlands region, so cover a lot of areas around Birmingham and the Black Country and places like Coventry. And our focus really is to, canals are very accessible to the public. They provide great nature on people's doorsteps, but also we can include a huge amount of community engagement activities, as well as providing interconnected habitat across a vast area of the network. So we pride ourselves on having an asset that's freely available to the public and provides a lot of opportunities for local communities to use and enjoy. So that's just a rough background about the trust. But with such an accessible network, obviously there comes issues with invasives, especially in terms of the pervasiveness and the ability to spread. So canal environments are somewhat unique, but they do have a number of issues that we have to address. So just a background about the project, really. CRISEP is, as Callum said, we have got eradication involved in the title, but it's very much is a bold statement. And we're sort of, the project overall is trying to look at areas where we can significantly reduce existing populations of target inns, but there are areas where we do feel that we have very much good scope to deliver localized eradications, especially on the aquatic side. And we're really sort of developing management plans for individual sites and trying to break the overall catchment down into target site-specific areas for treatment and utilising different treatment methodologies depending on the site conditions, whether that be hydrological connections with rivers and the like. So it's fully funded by Severn Trent Water as part of their Great Big Nature Boost scheme. It's a 4-year project and it's around £600,000 worth of funding. And the 4 species we're really tackling is Japanese knotweed, giant hogweed being the 2 terrestrial, and the 2 aquatic being floating pennywort and floating water fern. So today I'm obviously going to just focus on the latter 2, but overall, really, the Severn Trent catchment supply area is a huge, vast area, as you can see on the map on the post on the right-hand side. And we're targeting around 250 kilometre lengths of canal. So there could be a kilometre length of pennywort, or there could be a site with just a couple of stands of Japanese knotweed, but overall we're covering it over sort of a 250 kilometre area. So it's fairly fast, and specifically on canals, some sections of rivers and reservoirs as well. So yeah. So in terms of the aquatic inns on our network, really, we do have a number of problematic inns, and canals are obviously fairly unique, very different hydrologically to rivers. But also we have the added complications in terms of things like navigation that can obviously cause the spread of certain invasive species, and the fact that our network is so accessible to everybody. Millions of people have direct access to our canals. So there is always risks of introductions from fly tip material and waste and things like that. So we have to be very mindful that there's multiple pathways that can cause the spread of these INS across our network. And from the aquatic side, it can really range from having floating pennywort issues in canals, and to non-native waterweeds like Nuttall's waterweed and Canadian waterweed in terms of some of our reservoirs as well. And this can obviously cause a lot of issues for fisheries and sometimes sailing clubs that lease out our land and our reservoirs. And obviously, as Emily mentioned in a previous talk, these species can cause a myriad of issues, really. For us, from the aquatic side, navigational restrictions is a real big concern. Obviously, we have a vast customer base focused on boaters, whether that be residential boaters who live on the barges, or whether it be from people going on holidays and doing 1 or 2 week tours along our network. And some of these species obviously can provide real problems when we're navigating busy stretches of canal. And also there's concerns around water supply issues as well. So we heavily rely on around a significant number of reservoirs to feed our canal network. A lot of them are specifically built for canal feed, but in some areas where we have feeder channels, we have aquatic invasives, and obviously that can restrict the flow, the discharge volumes that can enter the canals, and it can also cause sedimentation build-up, where we then have to look to dredge some of our feeder channels to improve the overall water resources supply to some of our major canals, such as the Worcester and Birmingham Canal and other parts of the BCN. And obviously the impacts on native flora and fauna, as we're all aware. So, as I said, we're trying to achieve significant specific site reductions, mainly on the aquatic side. The terrestrial side is obviously much more challenging for us, but we do feel like we could have some serious success, and I will be showing you a number of methods that we're using to really tackle these individual plants. And just to show you as well, we developed a new GIS treatment survey so that we can accurately map and geo-reference all of our treatments. The contractors can use GIS so that not only can we plot the points of where we're doing the treatment, but also we can have a data package for each individual site. And there's hundreds of sites, including the terrestrial side. But then we can get the exact locations, the quantity and the volume of product applied, if it's chemical, or the number of weevils applied, what areas this is covering, and also the site-specific fixed point photography, so we can see the temporal change over time to really understand on a much more frequent basis how effective some of our controls are doing, especially on the biocontrol side. So first up, floating pennywort. With regards to this species, this can be quite a difficult one for us at the Canal River Trust. Obviously, canals are very low flow in comparison to rivers, but a big issue for us is the fact that our canals accumulate a lot of sediment and silt, and there's a nutrient response and a causal issue with regards to the ability of floating pennywort to harvest nutrients, even within sediment in areas where we have significant sedimentation and very high organics and nitrogen and total phosphorus. So you'll see a running theme throughout this section of how we've sort of developed a synergy with our dredging projects and how we tackle invasives as well. So it's quite an interesting way to allocate resources and finances so that we can tackle two issues at the same time. But specifically in year one, over the growing season this year, we've targeted 4 priority areas in the West Midlands, and I've put a cheeky little floating pennywort icon on the map, so to represent those locations. But we're going to cover 4 of those, and I'm going to go into a bit more detail about some of them on the next few slides. So we've got a mix between canals and feeder channels from reservoirs, and some of the canals, sadly, we do find pennywort on our triple SIs and SACs, and which can contribute to the unfavourable conditions. So by hopefully delivering this project and getting some successful outputs, we can improve some of the condition statuses of our protected sites. So first up was the Fens Branch Canal down in Stourbridge, and this is actually adjacent to a really important site of ours, the Fens Pools triple SI SAC, really incredible site in a very urbanised context. But the pools there actually feed down into the Fens Branch, which ultimately ends up in the Stourbridge Canal. And we've always had issues with pennywort here. You know, we've tried a number of different techniques. We've also tried to have sort of different contractors involved to really try and see if there's any improvement in terms of quality of works delivered by our framework contractors. But this year we wanted to really tackle it using mechanical means. We did use some chemical, and in some instances we will have to as a follow-up, but we would never just go and spray glyphosate as just a single treatment method. And in many ways, I find it very ineffective. There's a lot of site conditions that you need to consider. But in this case, we was really concerned because we obviously get water supply coming into the canal via the branch. But also it's quite important for biodiversity as we're so close to such an important European designated site, as well as its connections with some of the other major canals in the western part of the BCM. So we used a conver boat. So this is a type of mechanical craft where we can, it's very maneuverable. It's very easy to change direction and adjust to flows. It's a very nice piece of kit from one of our contractors, but it's really useful because we can use different attachments on the craft to remove the pennywort. So sometimes you may want to use cutters, sometimes you may want to use aquatic weed harvesters, but you can adapt it to the situation that you're in, especially if you want to get into some of the very dense areas where the rooting mass is substantial. We're talking, upwards of a number of tons really in some of these areas by the time that we've moved it. But we had a very strict biosecurity regime in terms of booming off sections of areas, having bubble curtains installed as well to prevent any floating matter migrating to other sections of canal. And as you can see on the top image, we had a hopper that was moored very close to the area of treatment so that we could remove this, and it was ultimately used at a composting facility that can be used potentially for agricultural benefit after decomposition as well. But the spot chemical treatment which did follow, it was around 5 weeks after where we introduced glyphosate, but we used a specific adjuvant being codicide oil because some of the research suggests that top film as an adjuvant is only useful in the early stages of the season, whereas this was fairly later, post-June. So codicide oil is a great way to ensure that the glyphosate is not just running off and ultimately causing pollution. And it almost binds the plant much more effectively using a type of adjuvant. If we just spray glyphosate for the sake of it, we have to think about the conditions as well, ensuring that there's no rainfall pre and post works to ensure that the glyphosate and the codicide oil effectively works at binding to the plant and attacking it. Here's I love to have some photographs. I think with invasives, it's always good to see pre and post. With things like Japanese knotweed, sometimes you see a dead plant and you think, oh, a lot of members of the public think, oh, what great, it's died, it's not going to come back. And then by the time it's the late spring of Japanese knotweed has re-established very well, and it can take a very long time. But from the aquatic side, we can see real changes. So this is the Fenn's branch. You can see how dense the pennywort was pre-works, and this was about a month and a half after we delivered the mechanical and then started the chemical works. And we'll continue a process of that over time, but also we can introduce volunteers who can maybe help remove any re-emergence. And over the next few years, using that and the spot chemical methods, I really believe that we can be successful at this specific site. Here's another site, Jacob's Feeder, which is, as I said, a feeder channel from the Bittle Reservoirs SSSI. This one was really important from a water resources perspective because the channel directly feeds the Worcester and Birmingham, the Upper Bittle Reservoir. If you've never been there, it's an incredible site for biodiversity. But this one was in combination with a feeder dredging project. So not only are we removing, from the first phase, removing the pennywort as best as practically possible, but we're also removing a significant amount of nutrient mass as well. And in this specific sediment, the total nitrogen and total phosphorus percentage volume concentrations were very high. So of course, that's great because pennywort in some areas can be used on wastewater treatment plants for harvesting nutrients. It's very effective at doing so. But in the long term, we can get rid of that substrate and also remove as much of the nutrient feed that the pennywort can potentially be tapping into. And also we used a lot of that material on adjacent landowners, so we could actually use it as an agricultural benefit to improve soil fertility on adjacent land and create bunds to prevent any soil erosion taking place over the winter period as well. So that was end of life solution there, to coin a phrase from this sort of circular economy perspective, really trying to make use of the material. And also the pennywort potentially would have improved water quality in the local area by reducing nitrate concentrations as well. So it does have its benefits, but ultimately, from our perspective, more problems than benefits. Again, strict mitigation. The interesting thing from the water quality perspective, we did undertake a fish rescue before we did this work. We actually removed 2 eels from the feeder channel as well. But there was a huge rapid increase in DO after we removed the floating Pennywort, especially before the pre-dredge, because the sediment obviously wasn't in flux and we was getting a lot more sunlight into the channel and removing a dense mass that was potentially restricting some of the other plants within the feeder. And obviously we took samples for cabby, as part of their study of looking at different populations across the UK. Here's just a satellite sort of image. This is the Cannock Extension SSSI SAC, again, just showing you the type of square meterage in terms of the Pennywort. Not hugely pervasive, but it's very dynamic. It varies year on year, of course, but at the top end, where there's a specific 350 square meter mass, I think we have something like 8 cube per meter of sediment as well, and that's where the huge amount of mass. So there's a direct correlation between quantities of sediment and the success of floating Pennywort in terms of its establishment. And this is a dead-on canal as well, so presenting some more issues there. So there's just an overall image of some of that. So moving on from that, we're going to move on to water fern, another one which is very problematic. I believe it originated in got introduced into the UK via horticultural means, I think in the 1920s, and I believe there was some research done on that, but this in the canal environment is a very problematic species and very different in terms of its spread and its flux year on year, really, in the canal environment in comparison to Pennywort. Not so much an issue for navigation, but from a biodiversity and an aesthetic perspective, especially health and safety as well, from people thinking it's grass when it's green in the summer. For us, from a perspective of that and public safety, obviously it can provide a lot of issues. So I've plotted some nice images of weevils on some of the canals where we've introduced a biocontrol in partnership with Cabby. But in this specific year, we focused on 3 priority sites very close to one another in the West Midlands, where we did a number of targeted releases. But this, again, as I said, this does change year on year, and sometimes the azolla is out competing duckweed, and sometimes the duckweed actually, strangely enough, out competes the azolla. So there could be an interest in a bit of study that we could do in terms of changes in nutrient concentrations and how that affects the pervasiveness of different competing species as well. But yeah, predominantly the Northern BCN, Birmingham Canal navigations, being the Walsall Canal, the Worley and Essington, and the Doe End as well. Really important from a biodiversity perspective, because there's a number of SSSIs that are interconnected with our network along this length. So yeah, as I said, we use a biological control thanks to Cabby. Cabby have been doing this for a number of years. It's been licensed a lot longer. So, lots of different organisations use these specific weevils. But obviously canal environments do pose somewhat of a different challenge because they're linear corridors, and we have navigation that can influence the spread of certain species as well. But we released just under, around 6,500 of these weevils across those priority sites. And we've mapped these using our GIS survey that we developed in-house. But interestingly, we know that obviously azolla can take nitrogen from the atmosphere, but we're starting to see some interesting correlations between surface water discharge inputs into our canals and the pervasiveness of certain aquatic species. And in this area, we've got a very high density of surface water outfall discharges into our canals from a number of different occupiers and different assets. So, apologies. So we've been getting help from the University of Manchester and their biosciences department in terms of doing some of the monitoring and having releases supported by some of the university students. And we're monitoring these using photographic evidence as well as doing some water quality investigations to see how certain physicochemical parameters change over time with changing masses of azolla once we know that the weevils are taking effect. And in 2022, to take this a bit further, I would like to do some aerial drone surveying so we can survey much longer lengths rather than doing ground truthing. We can have sort of drones surveying much further lengths as well, rather than us and myself having to walk long lengths of canal. Why not use modern day technology to do so? So there we go. So Interestingly, this year we had a very late bloom season in the water fern. It wasn't really established as we've typically seen sort of in the late spring, early summer. It wasn't till around September when we saw these mass blooms that happened really quite rapidly. And obviously that provides concerns in terms of the viability of the weevils to breed, for I guess from Cabby's perspective as well, but also the predating success and how that changes as temperatures decrease. Will weevils overwinter? And there is some suggestion that in some areas they have, in some areas they haven't. So it was much more difficult that we had a smaller time frame in order to do the biocontrol. As you can see, the release dates on the right within the September period. But some areas we've seen huge successes, over sort of 90–95% reduction on site-specific areas. But then in other areas like the Worley-Essendon Canal, we've seen very minimal changes as well. So there's obviously a number of site conditions that can influence this, as seen on the previous presentation with the Rush trial. We have to take into a lot of account of the sediment fluxes in terms of nutrients, as well as water quality, atmospheric deposition of certain chemical parameters as well. There could be a number of variables that are influencing this, but as I said, surface water outfalls density tends to correlate with areas where we're finding these significant infestations. Here's a nice before and after photo. As I said, I do love these. So this was one specific site on the Doe End. Doe End has been one of the most successful biocontrolled projects this year. At Parkline Pits Local Nature Reserve, this area you see here, we've seen just over 95% reduction, and I'm still monitoring these, even though it's coming into the winter time and we're going to start seeing some of the marginal dieback. Significant within 4 to 5 weeks. This location, and I've walked kilometers of canal to survey the rest of the area to ensure that wind direction or flow hasn't had an influence in terms of breaking it up into smaller chunks. Completely across this length of canal, all the way south to the next canal where we actually do have a lock flight, there was almost complete 100% reduction in the Zoller infestation in a 5-week period. It was actually quite rapid, but in other areas we saw 30 to 40%, and in the Worley-Essendon there was minimal change. So as I said, looking at the environmental monitoring aspects in terms of DO changes, pH, but also we may need to introduce some other parameters to look at to see what is affecting the success of weevils in certain areas. And I'm sure the Cabby scientists can provide a lot more detailed data on that. So in terms of research as well, just to follow on this, I'm sort of more from the environmental chemistry background. So I'm interested in environmental conditions and how that can influence certain invasive species. So one thing that we need to ask ourselves is, how much influence does nutrient enrichment in these systems help with establishment? Well, not as much establishment, but the pervasiveness of some of these aquatic invasives. We know that there's some areas, some reservoirs where we know we've got high phosphate concentrations, which and significant issues with certain aquatics such as the zoller and the least duckweed as well. But we really need to understand the nutrient budgets within these systems as well. How does sediment fluxes increase phosphate within the water column? How does atmospheric deposition influence that via rain gauge monitoring and sampling? So, and what is the nutrient uptake as well for the different organisms? So we're going to work with some interesting consultants on this, and also not only to determine the sources of inputs, but also to understand, is there other techniques that we can use, which I'm going to show next, for tackling the environmental conditions on the site, which are ultimately leading to the issues that we're seeing with certain aquatic invasives. A more holistic scientific approach in terms of managing these long term. And just to mention on that last point, we are in talks with Phoslock as well, because some of the areas where we do have high phosphates, we may potentially be able to use lanthanum, which is a rare earth metal company called Phoslock that has developed this, which is mixed with bentonite clay, to reduce the phosphorus concentrations in the water, but also provide a capping layer in areas where there's a lot of sediment, and figure out what are the cyclical timescales in terms of phosphorus release. And when it does, could the Phoslock and the lanthanum take up and lock a lot of this phosphorus away and give us a chance to maybe introduce new macrophytes or tackle a lot of the water quality issues that are causing some of the concerns. And the site that you see on the bottom image there has had a lot of issues with sewage inputs and the like, and it does have a feeder brook that goes into it. So we need to understand how all these different inputs are influencing the development. As you can see, it's almost completely covered in green from the azolla and the least duckweed. And is there a way that we can introduce certain things into the environment, such as your dosing or whatever it may be, such as Phoslock, to really tackle those aspects as a restoration to tackling some of the issues what we're seeing with the invasives. And another one, just to mention, we're currently in discussions with Cabby, potentially next year having a floating Pennywort biocontrol using another weevil, much bigger than the one that the North American one we use for Azolla. But this will be the first time it's going to be released next year in the experimental trials. So we're currently in conversation with Cabby at the moment. So hopefully we'll be able to provide some funding for this and be the first time it's done on inland waterways, and we'll compare the effectiveness of this control with some of our mechanical methods as well. And lastly, as an additional, just to add on, Himalayan Balsam, this is something I don't believe has ever been trialled before. We don't really tackle Himalayan Balsam on a catchment scale. We do have local action groups and volunteers, but in terms of eradication, it's not something that's a focus on the project. But we are dredging a feeder channel, Pennar feeder, which goes into the Montgomery Canal, which is a triple SI and SAC. We've got about 400-meter length of Himalayan Balsam on the offside bank of the feeder. So what we've discussed is, how about we trial something that's never been trialed before, and it may completely fail, but why not use the dredgings from our dredging project to try and suppress the growth of Himalayan Balsam, provide a biodegradable jute netting on top of this to add even more suppression, and then hydroseed sediment and soil on top of that layer to provide a third step in a tier of substrate with native species at a higher mix in terms of the ratio, to see if we can provide enough suppression using natural materials to then allow the establishment of native species. The one thing that we have to consider is there viable seeds within the feeder channel that have ultimately ended up in the seed? So we'll have to do a number of sampling to see that. I don't think it will, because of the flows within the feeder. They're more likely to drop out into the Montgomery, but it's something that's, as far as I'm aware, it's never been trialled, and it's very site specific on a much localized scale, and we'll be able to map the changes over time. You know, that we may get some success, or it may completely fail, but you know, we need to try this. That's what innovation is, isn't it? At the end of the day, we need to trial new techniques, and ultimately, if we can suppress the Himalayan Balsam, we'll be able to reduce the future dredging costs, because when we get the winter dieback, we get significant soil erosion, which ends up in the feeder channel. We then have to spend 80 to 100,000 pounds dredging it on a cyclical basis. If we can suppress Himalayan Balsam, maybe we can reduce those soil erosion inputs coming into our feeders. So that's it from me. Thank you very much. And if you've got any questions, please feel free to fire away. Thank you.
Thanks very much, Charles. Whistle Stop Tour, they've been a busy boy in year one of that project. Really impressive stuff. A couple of observations from me. I'd really want one of these little conver boats, please. They just look like the business. Could do some damage with those. We've got a couple of questions, particularly in the Azolla thing, I think. Maybe first one is from Colin, and it may have been partly answered in the chat room earlier, but do the Azolla weevils need to be released every year, or are you looking to establish a self-sustaining population where you've released them? I think there's maybe a suspicion from Andrew Skinner's comments earlier that they do establish another winter, so I presume the answer is that you're hoping once you've released them once, they're there to stay.
Yeah, so again, it comes down to I think the site-specific conditions. Again, I mean, temperature obviously has a massive impact in that. If we get a severely long, cold winter period, then you know the chances of overwintering probably reduces substantially. I'm sure Cabby have got more data on that, but there has been areas where we have introduced and we've not had to do any top-ups, and it's controlled the Azolla really well, especially I think around 3 years ago in another part on the western area of the BCN. They introduced weevils, and one season, and it seemed to suppress very well, and we've not had issues since. However, when you've got major infestations like we do now on the northern BCN, it's highly likely that we're going to have to do a number of top-ups each season. But we could potentially do some areas where we could do mechanical control. Mechanical is not great for Azolla because it can spread via its fronds, and then you may be actually spreading it around a lot easier. But I think in these specific areas, we're more likely to have to do a number of releases, but we will see next season to see how effective they have been at overwintering.
I had a question, Charles, myself actually. Is there any sort of steer as to, what's your density of weevils that you need to release? I think you mentioned somewhere about 6,500 had been put out, and you got different sort of responses in some of the different sites. Is there a weevil per metre squared rate that you need to be working to, or do you just put them in there and they replicate themselves within whatever situation you put them into?
Yeah, there is usually a specific rate, but I can't remember. On the Cavi website, I'll have it. I can't remember the exact rates, but what we did was, because obviously we had such a limited window, we would have probably introduced a lot more than we did because the infestations were vast. But in the Doe End example that I showed, they were typically sections of a few hundred square meters. So they were much more localized, whereas on the Worley and Essington Canal, we had bank-to-bank infestations of kilometre lengths. And we could only introduce, in the time frame we had, we could only introduce a couple of thousand in that area. So there was some limited reductions, but I believe if we was to try and really scale that up, we may be in the tens of thousands weevil. So we'll have a whole weevil army for some of these sections. But as I said, nutrient inputs and other factors need to be considered and need to be understood as well, and how that can influence establishment.
Okay. I'm going to pick one of Gideon's questions here about, you mentioned duckweed potentially competing with the Azolla, and the question is, have you found if white or yellow water lilies can compete with Pennywort, or is that something you've seen or observed as yet?
So we do have a lot of, we get a lot of lilies on the canals, but sadly, in all the areas that I've seen of Pennywort, there is pretty much, it's almost a monoculture of Pennywort, really. It outcompetes everything that I see in the specific sites that I've looked at. I mean, on the Cannock extension, we've got a very important protected species, Loronium natans, floating water plantain, huge beds of that. And sadly, the Pennywort has spread a vast area, and there is concerns that a designated species such as that could be negatively impacted from the Pennywort. But I mean, in a canal setting, it's obviously it's quite changeable, but in all these sites that I've looked at, Pennywort tends to just completely take over.
Yeah. Okay. Gideon's got another question about balsam, but I'll maybe ask you to deal with that one on the chat. Perhaps I don't want to go back to balsam. We've done balsam today. So, Charles, I think maybe what we'll get you to do is have a look in the chat room as any of the other questions come through, and we'll let you respond to those directly. In the Scottish context, we've not got a vast canal network, but clearly in your neck of the woods there is, and of course there's a sort of, the legend, which is, there's more canals in Birmingham than there is in Venice, and that gives a sort of scale to the problem that you're dealing with and a scale to the project challenges. So, we wish you really the best of that, and we'll love to keep up to date with what you're doing in the next few years of your project, so good luck with it. Okay, without further ado though, what I'm going to do, I'm invite Charles to mute and hide his face for a bit, and I'm going to introduce our final speaker this afternoon. Karen Muller is one of the project officers for the Scottish Invasive Species Initiative. She looks demure and calm at the minute, but trouble on legs is our Karen, but she's also our very own sheep whisperer, and she manages our project working in the River Devon catchment, where we're looking at using sheep as a grazing tool to control giant hogweed. This always gets a bit of interest when there's videos posted online, and Karen's our sort of media darling, because there's always some sort of radio presenter or whatever wants to speak about it. But we did actually, want to sort of share a little bit as to what we have been doing with this work. We're using sheep as a grazing tool for giant hogweed management. Sort of, give a little bit of a feel as to how that goes and potentially where it might go next. So Karen, we'll give you a chance to share your screen and come off mute, and I shall hide my face and I shall go on mute. Hopefully you're going to appear in a second. Yep, we got you, Karen, that's it.
Excellent. Well, as always, I'm very excited to introduce to you all our flock of hogweed munching sheep that we're using in the Devon catchment. So without much further ado, I will let them flock to the rescue for giant hogweed control. So we have been running this trial at the Macduff site since 2019. So we've just done our third year. And what we're trying to find out really is, can land managers use sheep to control giant hogweed? And best case scenario, also how can they do that best? So what we're trying to do is establish an optimal grazing regime, and then based on our trial that's been going for a few years now, and then produce a guidance document at the end of our trial. So that will hopefully be coming out early next year, which will give an opportunity to give land managers or anybody else who might be interested to use sheep as an alternative a chance to learn from the lessons that we've learned and the info that we can share. So if you want to read up a little bit more about it, I have included a link on this slide here. Our case study on the website is updated annually as and when the results come in. So if you wanted to read a little bit more about it, that should be the place to go. As a little bit of background, the Macduff sheep grazing site is right at the bottom of the River Devon. It's a strip of mature woodland that's kind of right wedged in between arable land on one end, and then on the other side is the River Devon that flows along. And there's a number of streams that run through the site. So really quite nice conditions for giant hogweed to have really gotten established. It also is a popular recreational route. So there's a footpath going through it that is quite well attended by walkers and dog walkers. And as you can see in this picture, there is quite a significant giant hogweed infestation that's very dense. So again, that also with recreational use in that area also means that there's a public health hazard, as well as for dogs that are walking there. The previous control that has been going on there was really just chemical control. And what we were finding was that it was incredibly time consuming. It was really difficult because of the terrain at times, but it was also pretty ineffective and very expensive. So the farmer was a very keen bean to get on board with this and to help us out with this flock of sheep to see what we can do. As just a little bit of background, because most people will probably know about giant hogweed at this point, it's got all of the really common threats that invasive species, invasive plants, especially in riparian habitats, pose, outcompeting native species, dying back in the winter, so really contributing to bank erosion, and then obviously an increased sediment load in the river, which impacts on invertebrates and juvenile fish, and also, of course, with the giant hogweed, the phototoxic sap, which causes burns and blisters on the skin. But for the alternative method of using sheep for control, some of the most important bits to remember here are actually how giant hogweed grows and behaves. So a giant hogweed from seedling to flowering can take a number of years. What happens is that it emerges as a seedling, it will grow for a season, it'll start storing up energy in its taproot, die back in the winter, and come back the next year as a bigger plant with more energy stored over that next growing season. And it will continue doing that depending on conditions that can take a varying length of times, but it will do that until it has stored up enough energy in the taproot so it can flower and go to seed, and then it will die and not come back. And once it has gone to seed, each plant actually produces around 20 to 50,000 seeds, which are then viable for 3 or more years in the soil. So this is really important to remember because it's the introduction to sheep aren't miracle workers. What we're looking for the sheep to do is, in terms of seedlings, that can be pretty much eradication because the sheep will go over it once. If it's very, very small seedlings, like you can see in that very first picture I've got here, and they probably don't have any energy stored up. So the sheep graze it and the seedlings are gone. When we're coming to these slightly larger plants, they actually will have energy stored up. And so it takes the sheep to go over these areas. They will eat the foliage and hopefully some of the stem, and then it will force that plant to use energy that it's already got stored up to push more foliage out. And basically the sheep are continuously working to graze that down, deplete that energy until that plant is dead. This is really important to remember as we're going forward in this. So the next thing I've got is a nice little trail cam video that was taken over a number of days on our site, just for some visual impact of how sheep interact with giant hogweed. So this area you can see is pretty densely infested by slightly larger plants, and you can have a little preview of the sheep munching away at this now. You can see that's already reduced and it's brought its friends in. And this is just a few days later when they've been tackling it quite nicely already. And then as usual, the battery in the trail cam died. But when I did return to that site, there was just stalks left in that area. But we're coming right back here to the sheep are grazing the leaves, but these plants will come back and the sheep will have to go over it at least one more time, probably a couple more times to really kill these plants off at this stage of their growing. It's not really a secret that sheep will eat giant hogweed. So monitoring of this trial site is actually really, really important to answer some questions, burning questions that we all have. We're doing this monitoring together with the University of Aberdeen, and we are monitoring both the grazing impact on the volume of giant hogweed across the site, which are these 42 little blue plots that you can see in the map. And we're also monitoring the grazing impact on the rest of the vegetation through habitat and vegetation monitoring, which is happening at these 7 orange points across the site. This is really important because we need to know that, first of all, how effectively are sheep tackling the giant hogweed, but also what sort of impact are they causing on the rest of the vegetation? Because while we would like to get rid of the giant hogweed, we also want to conserve the rest of the native vegetation that is on that site. So the pictures at the bottom give a pretty nice little overview of 2019 and 2020. So the very first one was taken in June 2019. That was the very first year that the sheep were released onto that site. And you can see that there was a lot of hogweed. And when we returned in October in 2019, that scene looks vastly different from the sheep grazing. They really just razed it to the ground. Shortly after the sheep were taken off the site, and then they were reintroduced again in the spring of 2020. And in June, you can see that that is already also a vastly different story to June 2019. Though most of the green things that you can see popping up there on the ground is actually giant hogweed seedlings. But as we revisited that exact spot in September 2020, they again had really raised that to the ground. So what have we found out so far? In 2019, we introduced 25 sheep onto the site for the first time. And these sheep had never really encountered anything like giant hogweed. So they were really reluctant to eat it. They were sort of just hanging out in the grassy areas. And we actually had to pen them in for a little while in an area where there was mostly giant hogweed for them to develop a taste for it, which, to be fair to them, they did fairly shortly after. And then we released them back onto the entire site and they started tackling giant hogweed really effectively. So we did find in 2019, giant hogweed control by sheep was great, really effective. But what we're also finding is that there was a huge amount of overgrazing of other vegetation. And also because of heavy grazing and sheep usage, we were also having poaching of the ground, so creating areas of bare ground, which again, obviously isn't ideal. First of all, you're preparing the ground massively for the next seedlings to come through, but also we want native vegetation to be conserved. And we also wanted to, at least in the future at some point when we've sort of gotten a little bit more on top of the giant hogweed, to help outcompete any seedlings that are coming through. So in 2020, then we about halved our sheep grazing days, and we halved our number of sheep. So about 50% reduction in total sheep grazing days of that season. And what we found is that the sheep still really effectively tackled the giant hogweed. And there was a pretty big reduction in overgrazing that was going on, but there was still overgrazing going on. So in 2021, our recommendations were that we would further decrease these sheep grazing days on the site. So again, we started off with 12 sheep, and we reduced that to 1,320 total sheep grazing days. Compared to 2019, that is actually nearly a 74% reduction of sheep grazing. We're waiting on the results currently, but from what it looks like so far, that looks like that's a pretty good grazing pressure for the site. We were finding that, again, hogweed tackled really effectively by the sheep, but overgrazing wasn't that apparent anymore. And that is exactly what we would like to do. Effective tackling of giant hogweed, but also either really significantly reducing, having very minor overgrazing impacts, or preferably avoid it entirely. What we have learned from this so far is actually that overgrazing, especially initial overgrazing, really alters the composition of the vegetation and the structure of the composition long term. So we would much prefer initial undergrazing to overgrazing, because you're monitoring and you can always increase it. If you come to the site in August and you think, these sheep need longer on this site, we haven't quite gotten to a point where we feel like we've tackled the giant hogweed effectively, then you can always leave them on for a little bit longer. It is much, much more difficult at that point when overgrazing has already happened, you've already altered the vegetation composition. It is much more difficult to reverse those impacts. You're going to be creating the spare ground, you're going to be overgrazing the native vegetation, and what you're likely going to do is replace, at least in some areas, native vegetation and also hogweed with things like nettles and thistles, these colonizing native species that are also really well defended. They're very unpalatable for sheep. And in some cases, we also found that it looks likely that the sheep aren't that keen to whack themselves through big swaths of thistles and nettles to go and get the giant hogweed that is growing amongst that. So to do us all a favor, it's always better to start low and build it up, depending on what the monitoring shows, than start really high and then have a situation where it's really hard to reverse that. The other thing that we found was that sheep will very nicely eat the smaller plants, but what we're also finding is actually that on the sites, the sheep were also feeding on the larger plants. In some instances, we even saw that sheep would rub against these big flowering plants and would knock them down and then graze them. So while we know that giant hogweed seedlings, especially, and also the other plants, come out and emerge in force in the early spring, it actually doesn't necessarily mean that it's absolutely necessary to put the sheep onto the site right then and there, because they will eat the larger plants as well. So I've got a nice little trail cam video here again of the sheep demonstrating eating some of the larger plants. You can really see it reaching up here and grazing that leaf down. So especially this year with the reduced number of sheep and with the reduced time that they were spending on the site, we got further and further into the season, and there was still areas where there was hogweed that had grown up quite a lot and it was very dense. And there was maybe on my part a little bit of worry if the sheep were going to get around to it, but there was still a few weeks left. And actually the next time I returned, they had really gone in and gone to town on those areas as well. So it really doesn't seem to matter if the giant hogweed is towering over them or not. They will still absolutely feed on them. As you can see, though, there is still some plants that have made it to flower. So we can't just wash our hands once we put the sheep on. They obviously need monitoring. The sheep themselves need monitoring, but also we need to at least go on once annually when things come into flower to make sure that we take our long-handled saws to cut down any flowering plants, because the sheep are very effective at grazing seedlings and staying on top of this emerging seed bank from the soil, and feeding on any larger plants that are already on the site. But the very last thing we will want in this instance is actually for anything to go to flower and set seed and add these thousands of seeds into the soil bank, because you're just prolonging how long you need these sheep on there. So it very much is something that will require some sort of effort from us annually to make sure that nothing does go to seed. Or also we found some giant hogweed, because it's fickle like that, growing up on trees. So checking these sites for areas where seeds might be coming from that the sheep really can't physically reach is really, really important to do at the same time. So those are the sort of takeaway points of start low, build your way up, monitor, and also make sure that you put in your effort to make sure that nothing goes to seed. This year, on top of that, we also started a seedling emergence study. The results aren't in from this yet, but this is quite an interesting little trial that we ran at the site, because what we know is that emergence period of giant hogweed seedlings can vary quite a lot between and across river catchments and geographic areas, depending on condition. So while we know that generally giant hogweed seedlings will emerge in a certain period of the year, it was actually quite important to have a look at when they would be emerging and at what time and in what mass they would be coming out. So what we're looking to do here is across the different river catchments, we established these 1 meter square plots along riverbanks where we knew that giant hogweed was present. And we cut down larger plants to ground level, and then pulled out and counted the seedlings on this plot. So this occurred all the way from March till October this year, 1 visit per month for each plot, and it is basically a grazing simulation. So by cutting larger plants back to ground level and by pulling out seedlings and counting them and recording that, we were basically pretending to be a sheep going over this area. So after each visit, after each month, you left this, at least in terms of giant hogweed, bare plot behind. And then you would leave it for a month and you would come back the next month and you would do the same thing again. You would pull out any seedlings that had emerged, and that pretty quickly gives you an emergence curve for giant hogweed seedlings over a number of areas. This is really important because it feeds into what we know about using sheep to graze giant hogweed. So our giant hogweed grazing trial gives us advice on the grazing pressure that's required. And our emergence curve from the seedling emergence study will actually help us define the optimal timing of grazing. So at what point is a good idea to put the sheep on, and at what point should the sheep probably come back off? So this is still in the works, but we're hoping that this might be a really interesting curve that will come out of this, that will really feed in meaningfully into our sheep grazing trial. And that concludes it for me, so I'd be happy to take questions.
Okay, thanks very much, Karen. We're really pleased with it, how the project's going, and as Karen said, we've kind of added it and refined it as we've gone. We've, like most projects, we've had to learn a lesson or two as we go, you know, particularly on the grazing pressure question, where we've really dialed it down. 3/4 of the grazing pressure is now removed this year compared to where we started, and I think, you know, that advice in there about, you know, start the pressure low and build it up is probably the key takeaway message at this point from the thing. We've got 2 or 3 questions, Karen. I'll fire them to you. Some of them you might have heard before, so I'll start with one of those. Paul's asking, was there any evidence that hogweed burned the nose or mouth of the sheep? And this is basically a pretty sensible and legitimate animal welfare question. So fire away, because I know we thought about this.
There was a previous sheep grazing trial that ran in the Devon catchment as well on a different site. And in that instance, actually, the sheep were extensively monitored by vets to look at exactly that. And in our experience, at least, both in the previous trial and in this trial, there has been no negative impacts on the sheep whatsoever from grazing giant hogweed. So in our instance, we can say that this has been quite safe and they're quite happy to eat it.
Yeah, Excellent. We've got a couple of questions about the sheep, Peter, and also Simon Cohen, about, you know, if we considered using other breeds like Shetlands or Hebridean, which are keener on browsing as opposed to prefer to graze. I don't know, you want to have first bat at that, Karen? I've got a couple of thoughts on it, but fire away.
So initially, the first trial started off with the Blackface Sheep, purely because darker pigmented Weheria breeds might be the first option to try, just because of potential hogweed burns. We have since moved on to sheep that aren't Blackface sheep with no issues, but at the moment, obviously these sheep are supplied by the farmer and they're managed by the farmer. So because we've had no issues, this is what we stuck with. And then obviously, again, monitor to make sure that they're not, or having as little negative impact on anything else as they can.
Yeah, I think what might happen from going forward, if this sort of approach was to be applied elsewhere, was you'd need to look at what the land manager actually wanted to put in place. We're kind of in partnership with the land manager here to persuade him to put some sheep on and take them off at our bidding, really, but we have allowed them to select sheep. But I guess there may be instances elsewhere where, you know, the land manager might have different breeds in preference. But what I would say is that most grazers will eat hogweed. You know, we've suggested that we should try goats and all sorts of different things. I think when you look across the literature, most of those sort of traditional grazers that we're familiar with will take hogweed. So it's more about, for us, trying to get the density of that, the pressure that we want that's going to deliver some sort of control. Alongside, as Karen mentioned at the end, you'll need to go and take the seed heads off at the end. So it's not a once and for all solution here, but actually over time it's a progressive reduction of the problem and removal of the seed count from the seed bed. Karen, there's a question though, which when you might, from Erin, which is about, do the sites with the overgrazing in 19 and 20, do they have more giant hogweed seedlings in 2021? I'm not sure what the answer to that is, but
we are still waiting for the results, but as far as I know, I don't think so. I believe from what I know from the giant hogweed monitoring plots that we've been looking at, the permanently marked ones that we've been looking at, it actually has been a pretty steady reduction on how many of these plots giant hogweed is found and how much hogweed is found. But that absolutely for this year still remains to be seen until we have the report in place.
Yeah, the sort of overgrazing that we're recording was a sort of generic overgrazing of the other vegetation that was there. And it wasn't a real response to see that they were being replaced by hogweed seedlings per se. Hogweed seedlings were in that soil bank and were appearing annually. But what we're finding was that once the sheep got in and done their stuff, they knocked over the hogweed and everything else. So we're trying to get the grazing pressure down here at a point where we can see some what is semi-natural regrowth in there. I think, you know, I've spent half my career sort of looking at grazing pressure on riversides and fencing off riversides with fishery-based organisations, and it's all about removing grazing pressure. So the trick here is to get the correct grazing pressure that allows the other things to thrive alongside solving or contributing to the solving of the hogweed problem. I think, as far as I can see, we've run out of questions. Oh. Hold on. Oh, Peter's got one about the golf club. I'll go for that. Can I ask how the golf club manage a giant hogweed? As assumed windblown seed gets on there, just in the context you said chemical control hasn't been very effective on our plots. Do you want to have first bash at that, Karen? The golf club up to?
I mean, as far as I know, the golf club does some giant hogweed control. I fill in the gaps there. The one saving grace is obviously that both the golf club on the other side and the sheep grazing trial are right at the bottom at the estuary of the Devon River. So whatever gets blown into the water, for example, goes out to sea. So there isn't a huge issue with infesting other unaffected areas as of yet. So
yeah, I think the ineffective chemical control was on this plot as well, on this location that Cam was talking about here. So it wasn't that they're saying chemical control doesn't work, because it actually does. but on this location where there had been previous efforts, they hadn't seemed to be effective. What I would point folk towards is, if you go on the CC website, if that's, I'm sure it's on your favourites list, there's a section on there on case studies under the plant control, and we're beginning to populate that at the minute with sort of individual write-ups of site-by-site examples of what we're achieving on a range of plants that we're targeting, including hogweed. So that's including sites where we are trying to tackle hogweed problem using chemical control, and we'll be able to show you know, the decline in effort, both chemical and time, that can be achieved if you're smart and strategic about control. So I think it might be worth having a wee look in there to see if there's any helpful support in there for you. I am conscious that we're running slightly over time, and the questions are pretty much finished for now. So what I'm going to try and do is wrap this up by saying a number of the usual things. Firstly, thank you very, very much to all the speakers, to Emily, to Charles and Karen, for giving up their time this afternoon, and also for the time it takes to prepare for these sort of things. It's a really difficult thing to talk to a laptop, not really knowing if your audience are alive, awake, asleep, or thoroughly enjoying it. But, you know, do it. This is how it is at the moment, and for us it's quite an effective way of getting 50 or 60 people to hear these sort of presentations. But thank you very much to the speakers for putting yourself through that. Thank you very much everyone for joining us, and again apologies for those that had problems getting in earlier on. We will have the whole thing recorded and up on the website, so you can revisit and put it on your favourites list forevermore. We have another one of these events coming up on the 18th of January, where we're thinking more about our furry friends, the mammals. The other part of the Scottish Invasive Species Initiative, other than plant control, is control of American mink. So we've got our next webinar is going to be dealing with American mink management from Xavier Lambin, red squirrel control in Wales, grey squirrel control, and also something about Scottish wildcat and the impact domestic cats on those. So, you know, if you're interested in those sort of topics, then you can book in for that pretty soon. We've not got the booking structures on the website at the minute, but Vicky is going to be sorting that out quite quickly. So thanks again, everyone, for coming along this afternoon. I hope there's been something a bit fun. We've tried to see the questions come through. Final thanks to my colleague Vicky Hilton, who is invisible here, but has done most of the grunt work here to get this thing set up, and including schmoozing speakers to get the presentations together. So without further ado, just leaves me to say thank you very much, everyone, and enjoy the rest of your afternoon. Thanks very much. Bye-bye.